Welcome to AP Biology. This video is a review of the cell cycle, mitosis, and meiosis. The cell cycle is all the events in the lifespan of a cell from the moment it was created through cell division until the moment that it itself divides. The cell cycle is made up of two large phases. One huge phase called interphase and a smaller phase called mitosis. Interphase is any time when the cell is not dividing. And during interphase, the DNA is going to be found within loose chromatin. During the process of mitosis, the chromatin is going to condense and form mitotic chromosomes. We'll be looking at the structure of chromatin in a minute. Interphase is made up of three smaller phases. You have G1, S, and G2. During G1, the cell is growing. It's also performing all of its normal processes. The S phase is when the cell really begins to prepare for the process of mitosis. During the S phase, all the DNA gets replicated. So you go from having single chromosomes to now having duplicated chromosomes that are made up of two sister chromatids attached in the middle by the centromere. During G2, the cell continues to grow and everything else in the cell doubles as it's preparing for the process of mitosis. If conditions are right, at the end of G2, the cell will enter mitosis. Mitosis is technically when the nucleus divides, and then cytokinesis is going to be when the rest of the cell divides, or the cytoplasm divides. The result of mitosis is two daughter cells that are genetically identical to the parent cell. Here we see the structure of chromatin. Chromatin is made up of DNA wrapped around proteins called histones. Together, the DNA plus the histone creates a nucleosome. And nucleosomes look like beads on a string. During interphase, we find loose open chromatin. You see it at this level right here. And you can see when you have loose open chromatin, all the DNA is accessible. So the genes can be transcribed and those proteins could be made. During mitosis, the chromatin is going to condense and it's going to form mitotic chromosomes. When we have mitotic chromosomes, the DNA is not accessible, so none of those genes can be transcribed during the process of mitosis. Proteins cannot be made using those genes. The cell cycle is very tightly regulated. There are three different checkpoints: the G1 checkpoint, the G2 checkpoint, and the M checkpoint. The checkpoints are going to be regulated using various chemical signals. Chemical signals tell the cell to transition from one stage or one phase to the next. The first checkpoint is called the G1 checkpoint, and the G1 checkpoint is really important. It's called the restriction point because that's when the cell needs to decide whether or not it's going to divide. If a cell is not going to divide, like a neuron or like a heart cell, the cell enters a separate stage called G0. The chemical signal that's used at the G1 checkpoint is the growth factor. The growth factor is an external signal, and this is the chemical signal that tells the cell to leave G1 and begin preparing for the process of mitosis. The next checkpoint is the G2 checkpoint. During the G2 checkpoint, the cell analyzes internal conditions, sees if the DNA has properly replicated, and if conditions are right, the cell could leave G2 and enter mitosis. The G2 checkpoint is regulated using internal signals. The major internal signal is a chemical called cyclin. Cyclin is a protein that fluctuates in concentration during the cell cycle. During the cell cycle, the amount of cyclin gradually builds up. When the amount of cyclin is at its height, it combines with another protein called a cyclin-dependent kinase, or a CDK. When the cyclin and the CDK combine together, they make a structure called an MPF, a maturation promoting factor. This MPF is the internal chemical signal that tells the cell to leave G2 and enter the process of mitosis. There's also a checkpoint right smack in the middle of mitosis during metaphase. And at the M checkpoint, the cell looks to see if the chromosomes are properly lined up. If at any point in the cell cycle, during the G1 checkpoint, the G2 checkpoint, or the M checkpoint, if the cell detects that things have gone wrong, if the cell has not properly replicated its DNA, if the chromosomes are not properly lined up, then apoptosis, programmed cell death, can be initiated. Apoptosis is an effective backup plan to make sure that cells that are wrong do not propagate themselves, do not have the ability to divide. Here we see the different stages of mitosis. It's not really important to know every single stage, but you have to understand the key ideas. Here you could see during interphase that the DNA is found within loose open chromatin. Here we see interphase on the top of the screen. You don't see the individual chromosomes. During prophase, the chromatin is going to condense and it's going to form these mitotic chromosomes. So the first time you could actually see chromosomes is during prophase. We can see that the mitotic spindle are forming. The mitotic spindle are made up of microtubules. They form from the centrosomes. You can see that prometaphase, the nucleus disappears. We can see that the mitotic spindle are attaching to the mitotic chromosomes, to these duplicated chromosomes. You can see that each duplicated chromosome is made up of two sister chromatids attached together at the centromere. The mitotic spindle attached to the chromosomes at a region called the kinetochore. During metaphase, all 46 chromosomes line up individually at the metaphase plate, at the equator of the cell. During anaphase, the sister chromatids migrate to either side of the cell. I know that we typically say that they get pulled to either side, but we know now that they actually walk to either side. Inside the kinetochore is a set of motor proteins. Motor proteins are powered by ATP, and the motor proteins walk the sister chromatids down the spindle fibers to either side of the cell. During telophase, the nucleus reappears. Cytokinesis, the cell splits in half. Animal cells split in half by creating a cleavage furrow. That's what you see in this picture right here. A cleavage furrow is formed by a ring of actin that gets wrapped around the cell and squeezes the cytoplasm in half. We learned that in plant cells this can't take place. Instead, a cell plate is formed. Basically, vesicles that contain pieces of cell wall are sent to the middle of the cell, and they actually build a new cell membrane and build a new cell wall, separating the one cell into two. Something that's really important to understand about mitosis is that the chromosome number stays the same. You begin with 46 chromosomes, you end with 46 chromosomes. The reason why the chromosome number stays the same is because the sister chromatids are being pulled apart. The moment the sister chromatids are pulled apart, they then become their own chromosomes. Now we look at meiosis. Meiosis only occurs in one part of the body. It only occurs in germline cells. cells found in the testes of a man and the ovaries of a woman. Every other cell within your body goes through the process of mitosis. Meiosis is going to have two goals. One, to cut the chromosome number in half and create haploid gametes. And two, to create genetic variation. We begin here during prophase 1. During prophase 1, this germline cell is diploid. All of its chromosomes exist in pairs. There are 23 homologous pairs. And to be homologous means that they have all the same genes in all the same places. You inherit one homolog from your mom, you inherit one homolog from your dad. You can see that during prophase 1, the homologous pairs come together, and prophase 1 is where crossing over takes place. We covered crossing over very well in class, but during crossing over, the homologous pair comes together and they overlap pieces of their chromosome. The spot where they overlap is called the chiasma, and the entire structure is referred to as a tetrad. When they cross over, they actually exchange pieces of chromosome, and that's going to function to mix up all the genes to increase the amount of genetic variation. During metaphase 1, the homologous pairs line up at the metaphase plate. Different than mitosis. Mitosis, they line up individually. During metaphase 1 of meiosis, they line up in their pairs. During anaphase 1, the homologous pairs separate from each other. And when they separate here is really where you see the effect of crossing over. They exchanged pieces of their chromosomes, increase in variation. Telophase 1, cytokinesis, the nuclei reforms. You can see that during meiosis 1, the chromosome number got cut in half. If we go back, we look in this original cell here, there are 4 chromosomes, or you'd have 46 chromosomes in our cells, 23 pairs. At the end of meiosis 1, you have 2 chromosomes in each cell, or you'd have 23 chromosomes in each of our cells. So during meiosis 1, the germline cell went from being diploid to now being haploid, having only one set of chromosomes. Meiosis 2 is identical to mitosis, except that it's happening with half the number of chromosomes. You can see that during meiosis 2, during metaphase 2, the chromosomes line up individually. During anaphase 2, the sister chromatids are going to be pulled apart. Telophase 2, cytokinesis, the nuclei reform, cell splits in half. During meiosis 2, the chromosome number remains the same. In this small example here, we began with a cell that had 4 chromosomes. We end with 4 daughter cells that each have 2. You can see that each daughter cell is genetically unique. Each one is different, and that's because of the crossing over that took place. Each daughter cell is haploid, has half the number of chromosomes. During meiosis, genetic variation gets created, and it gets created through a process called genetic recombination. Genetic recombination recombines or mixes up all of the alleles, all of the different genes, increasing genetic variation. There are 3 forms of genetic recombination that took place during meiosis. Number 1 was crossing over. Happens during prophase 1. Number 2 is independent assortment. Independent assortment of chromosomes takes place during metaphase 1, when the homologous pairs line up in the center of the cell. When the homologous pairs line up in the center of the cell, Nothing decides what side each homologous pair goes on. They could arrange themselves any way they want, and you can see this in this picture right here. The way that they arrange themselves during metaphase 1 is going to determine the combination of chromosomes, or the combination of alleles, that ends up present in the gametes. You can see that 2 homologous pairs can line up 4 different ways, 2 to the second power. 3 homologous pairs can line up 8 different ways, 2 to the third power. There are 23 pairs, so 2 to the 23rd power is an extremely large number. There are many different ways that the homologous pairs can line up during metaphase 1, and that creates a whole wide range of unique possibilities of chromosomes that can be present inside of the gametes. Independent assortment dramatically increases genetic variation. Finally, the last form of genetic recombination would be random fertilization. You have all these unique possibilities in the sperm, you have all these unique possibilities in the female egg, and nothing determines what sperm fertilizes what egg. It's all a chance event. Again, increasing genetic variation dramatically. That concludes this video.